All right, let's look at specific defenses or immunity. This is the resistance to the effects of a specific disease-causing agent, essentially our third line of defense. And what keys us off are these things called antigens. And this is a chemical substance that stimulates cells to produce antibodies. All right, these are found on the cell structures. Right, so we don't develop an immune response to the entire organism. We devise an immune response to particular parts of that organism, and that's what the antigen is. All right, and antigens are how we identify things that belong to us versus things that do not belong to us. So let's take a look at our immune response, and it starts right here with a macrophage. So what a macrophage is going to do is it's going to engulf a non-self antigen and the invader, that pathogen, whatever it is, a bacteria, fungus, virus, whatever it is. And then it's going to display that antigen there, all right? And from here, what that macrophage is going to do is it's going to move to a lymph node. And in that lymph node, it's going to encounter other lymphocytes and macrophages. So here it's displaying that antigen, all right, which is going to key off that immune response. Now, two types of cells here that are going to be involved. T lymphocytes, also known as T cells, and B lymphocytes, also known as B cells. So if we look at T lymphocytes, what T lymphocytes are going to do is they're going to directly attach to that foreign cell. Now the first cell I'm going to talk about are helper T cells. Helper T cells are going to stimulate B cells and they're going to stimulate B cells to produce antibodies. And so produce antibodies specific to that antigen. All right? They're also going to help these other cells called cytotoxic T cells. So cytotoxic T cells are going to attach to that non-self antigen and invader. And that's what you see right here. Here's a cytotoxic T cell attaching to that antigen and invader. It literally grabs onto it. And it's going to secrete these toxins called cytokines that are going to kill that pathogen. They literally poke a hole in the plasma membrane of that cell, and then the cytoplasm leaks out. So they're going to kill tumor cells, transplanted cells, foreign cells, and also virus-infected cells. So what's going to happen with a virus is that a virus uses our cells to make more of itself. So if we can kill that cell in that process, that's going to prevent less viruses from being made. So also, we're going to have memory T cells that are going to be made. Memory T cells are not going to respond to the first infection, but they're going to remember that antigen. So if we get exposed to that antigen again, they're going to provide a no-delay response to future infections. Next are B cells. So B cells is showing B cells here, all right? So from a signal from those helper T cells, those B cells are going to divide into plasma cells, and plasma cells are the cells that are going to release antibodies, all right, and also memory cells, so memory B cells. So once again, memory B cells, just like memory T cells, are going to attack subsequent exposures. Now I also want to point out that second exposure, our immune response is much more robust than the first time around. This is showing the first time around. Here's a B cell making a few memory cells and a few plasma cells, and here's our primary response to that. So we have a small number of these, all right? And so as you can see, it takes, anywhere from 3 to 14 days to get over that infection in most cases. Second time around, we get exposed to that same pathogen again. These memory cells were going to produce a lot more plasma cells the second time around, more memory cells as well. So our secondary response, the second time around, is much stronger than the first time around. So we're going to get over that infection much faster. All right? All right, so let's take a look at antibodies themselves. This is a picture of an antibody. They're these Y-shaped proteins. All right? So antibodies are also known as immunoglobulins, and these are proteins produced in response to a non-self antigen. All right? So let's look at how they work here. So this is showing the different kinds. So this is showing the modes of attack. One is that antibodies are going to directly attach to the antigen. So you see that here and here and here and all these other areas as well. And so what that's going to do, especially in this, it's going to bind those antigens together and the invaders together, and it's going to make it easier to phagocytize those pathogens. So a phagocytic cell here just has to do one phagocytic event to get all 4 of those pathogens, as opposed to having 4 separate events occurring. All right. Also, they can cover the toxic particles of the antigen. So sometimes, that antigen can be a poison to us, and so this is going to neutralize that toxin. They can also activate a set of proteins known as complement proteins, and that's what this is trying to suggest there. These complement proteins are going to attract phagocytic cells, they can alter viruses, and they can also cause inflammation. If you have my notes, I'm stopping here. We're not going to go and talk about types of immunoglobulins.